Welcome to session 3B of the PKDOC Summit. We're going to review a universal ADPKD protocol. So the purpose of this session is to develop a standardized ADPKD trial so that sponsors can go forward in the future with minimal effort and less regulatory uncertainty. Our objectives in the near term are to align trialist and regulator expectations for ADPKD trial best practices. In the long term, we want to support an umbrella program which reduces study time, cost, and patient risk. The methods that we utilized are focused on ADPKD endpoints, including surrogates, which are reasonably likely to predict clinical benefit, for example, TKV change, adverse outcomes, for example, death or end-stage kidney disease, function, such as GFR change, or symptoms, for example, patient-reported outcomes. Several academic and industry working groups identified challenges in applying such measures, their knowledge gaps discussing their use in accelerated or full approval. So we've consolidated this to provide a top-line summary, and regional regulatory representatives will provide their perspectives. Next slide, please. So the composition of the working groups are shown here. There were 3 90-minute sessions which were recorded, and the members of the different areas were shown, including a work group focused on cysts, labeled anti-cystic, and a work group focused on GFR, and a work group focused on others, such as measures of fibrosis, inflammation, pain, etc. The members of the different work groups are shown. Some of the individuals participated in more than one work group, hence the overlapping Venn diagrams. And the session leaders are shown on the right. The other session was led by Michelle Chunchel and Jalila McCauley. The anti-cystic work group was led by Alan Yu, and the GFR work group was led by York Pei and Leslie Inker. Next slide, please. So the endpoint working groups were asked to consider potential mechanisms to think about the study's population, to focus on the endpoint of interest, to discuss variables of interest, and look at measures to support interpretation. Next slide. So Frank, I'm handing it over to you at this point.
Thank you, Ron. Again, I'm Frank Czerwiec, and my task today is to go over the study design proposal that is a culmination of all of the work that these working groups came together with, and highlight some of the key points in which in subsequent slides we'll go into a little bit greater detail, but we're obviously not going to spend the 4.5 hours that were spent in discussion on developing this. So what you see in this slide is really an outline of what a typical ADPKD trial aimed at registrational approval might look like. The objectives are typically to study the effects or effectiveness of a chronic therapy on ADPKD progression while monitoring long-term safety. The working groups agreed in general that kidney volume and GFR are likely to be primary endpoints for the study. And we'll go over those endpoints in much more detail later. But that other endpoints like PROs and other assessments are not yet ready for prime time. Qualification or validation is still outstanding for a number of the instruments that might be used, but we agree that they do have a place in these trials and should be evaluated to fully understand the patient's experience. With regard to the primary estimates then, we agreed that TKV and eGFR are likely to be best evaluated in a subgroup of patients that have rapid progression. And what that means for the trial that we're proposing is that while you may enroll patients that have greater and lesser degrees of progression, you would define in the analysis plan that patients reaching some inclusion criteria would be the patients that are used for primary efficacy analysis. Information may still be gathered in other patients with lesser degrees of GFR decline or slower degrees or slower projected TKV growth. The population generally will be young, and most studies will start at 18 to 50-year-old patients because PKD really manifests its later end stages beyond 50 in the most rapidly progressing patients. But it likely will be broader. In other words, if possible, pediatric patients should be included in the phase 3 trials, and patients who are older may be included but may not be necessarily appropriate for analysis of the two primary endpoints. And again, CKD stages 2 to 4 are likely optimal for GFR efficacy, whereas Mayo class 1B to 1E might be evaluated for TKV efficacy. In fact, more likely you'll see studies that are focused on the 1C to 1E patients. The overall ends for the study are gross estimates, and this will obviously depend on the drug being studied and its likely efficacy. But we imagine that about 200 to 400 patients per group per endpoint might be needed for a number of 800 to 1,500 patients for a registrational program overall. And that can be done either in a single study where there's a study within a study, or potentially in two studies with confirmatory studies. And what we're proposing in this schema is actually a study that might lead to an accelerated approval based on an earlier readout. And that brings us to trial duration. Typically, each of these endpoints might require 1 year to 2 years of follow-up before you produce the formal analysis, formal statistical analysis to test the hypothesis. And typically this might be done for TKV first, which may have a greater degree of power if the investigational drug actually works through a cystic mechanism, and might be confirmed by analysis of eGFR or eGFR slope or some other measure of function with a longer time frame. So you may see an interim analysis, which is what we've highlighted with the red diamond, coming somewhere in the mid or early ranges of the study. Ideally, it will be a broad enrollment of patients with randomization and appropriate stratification across treatment and comparator. We will talk a little bit about standard of care and comparators in subsequent slides. But we believe that it will be important regardless to confirm an effect on GFR in later stage CKD patients, as well as explore the potential effects for GFR in earlier, younger patients, so that you can understand the potential for a therapy to affect the population over the whole spectrum of disease progression. We would encourage companies and others investigating investigational treatments to provide for an extension phase after the timed endpoints are reached, so that patients could be rolled over into active evaluation. And we hope that registries and other tools will facilitate confirmation of longer term ultimate outcomes such as ESKD and survival through this methodology. So I'll stop there and we'll move into some of the details of how we came up with this proposal. If I could have the next slide. One of the considerations, as Dr. Perrone had said, was to consider potential drug mechanisms. So we realized that drugs may target disease progression typically through slowing cystic expansion or possibly through direct preservation of GFR. But other endpoints can and should be measured, and we would hope that all of these would correlate with each other. In other words, we would see favorable effects for all, and that would be actually quite beneficial. Because there are potential for chronic and acute effects even with TKV, we think that the TKV can be used to study early disease, but will require a long period of observation. But other effects of drugs, such as effects on chronic activities like inflammation and fibrosis, may also be observed and may be beneficial in later stage disease. We agree and understand that the time to effect will vary by mechanism, and so longer observations are typically needed. Biomarkers could be particularly helpful in early stages of proof of concept to demonstrate that there's appropriate target engagement for the drugs and that they're doing what we hope them to do, and also to help with dose ranging and other important attributes. While we recognize that these acute effects on kidney volume or GFR are potentially likely for a number of medications, we also recognize that the study design is going to have to account for that and help the regulators in the interpretation of the overall chronic effects for each of these endpoints. And we think it's going to be important to understand as early as possible off-target effects that might complicate the assessment of these endpoints or might lead to treatment withdrawal. And it's very important to avoid missing data for long studies such as this, where endpoints require study visits, MRIs, blood draws, etc., to really avoid biases caused by missing data. So with we hope the regulatory concurrence, we're hoping that these investigational programs will study their acute or chronic effects of the drugs in early phase 2, that they will adopt appropriate measures and use estimands that will facilitate the integration or the imputation of data where appropriate and necessary. So moving on to study population, I'm going to just hit the high points for this. Subject number and power will typically be derived from the expected efficacy of the agent. Hopefully that will be estimated early in phase 2. But it will be important, and we would love to hear the various agencies' views on what they believe might be clinically relevant effect sizes for kidney volume or GFR. A number of proposals have been floated in the literature and at meetings, and what the ideal duration is needed and population size is needed for assessment of safety in the overall study, but also within important subpopulations. We believe it is important to study a range of disease stages. We had pediatricians involved in the working groups, and they really would like us to use endpoints like TKV to understand that a drug may have beneficial effects in patients that have not yet developed a decline in their GFR. And we know that regulators would love us to be able to study very late stage patients. And by doing this, we can really get a snapshot of the disease across its full spectrum and understand whether or not a drug might be beneficial. We believe that enrichment is important, stratification may be important across important covariates, and those all need to be accounted for in the study. There was a lot of discussion as well on the duration of the study and the potential need to use placebo in some studies. And while we agreed that ACEs and ARBs are a minimum standard of care for all patients who may be experiencing hypertension, we accepted that placebo might be able to be used for shorter durations to explore the effects of the drug outside of the other approved therapies for PKD. And it's possible that this might also be acceptable for populations where clinical equipoise does not require treatment as of yet, and we could use that to understand other patient populations. And despite the availability of a treatment for PKD, we understand and appreciate that not all patients can tolerate or use it. And we noted that tolvaptan should be made available for longer trials as a standard of care, but that if patients are not electing to use it or do not tolerate it, that those patients should be stratified equally across treatment groups so that you have an unbiased evaluation. Moving next, we're going to focus on the specific endpoints that the working groups targeted. I'll begin with GFR and kidney volume, and then Ron will pick up and discuss the other category. For GFR, we agreed that the typical end-stage kidney disease outcomes are really not useful for ADPKD, which is a lifelong condition beginning even before birth. But these outcomes might be helpful if there's a belief that the drug effects might persist into late disease and could be used for patients that are very near to ESKD and with a limit of time of follow-up. We hope that GFR can be used to sort of validate other endpoints like TKVs and PROs, and that a beneficial effect across all these measures will be useful in supporting a drug's utility, but also in cross-validating these various measures. We recognize that there are a large number of considerations for GFR measurement that must be addressed. There are a number of other consortia that have dealt with this in other forms of chronic kidney disease. And so we're not going to go into this in great detail. Suffice it to say that a number of methods for assessing chronic GFR slope or chronic change in GFR need to be incorporated and used to assess the true nature of a GFR effect. Now, we already mentioned that we need to avoid missing data, so the estimands should be constructed for that. That may include frequent evaluation, meaning you're measuring along various regular time points so that if a patient does become lost to follow-up, you would be able to take their last observation and use it in some way to account for that. Finally, and this really influenced, I think, the design that we are proposing. Various programs have already looked at PKD in either separate studies, typically, but they're looking at PKD at different stages. And we think that whatever program or programs are designed, whether they be single studies or multiple studies, you should be looking to see whether or not you can identify the different endpoints that are more meaningful for patients with early disease versus late disease. And of course, GFR is one that is more appropriate for patients that have already demonstrated that they are entering decline in GFR. So a combination of TKV and GFR as an example would be useful. And we believe that you could do this all within one study by enrolling a very broad spectrum of ADPKD patients, saying these are the patients that qualify for TKV endpoint, these are the patients that qualify for GFR endpoint, but we're going to study the effects of the drug across all patients as well to better understand how it might be used in general practice. In addition, we had a lot of discussion about GFR in early patients, particularly in pediatric patients with PKD, and we hope that there will be better biomarkers of functional decline that could be used in early stage disease. But we also hope that there will be regulatory flexibility to allow extrapolation of historical data or data that is obtained in run-in or comparison for early versus delayed treatment to help us to understand the longer-term effects of therapy and their long-term benefits. One such idea that is gaining some popularity is that patients can be tokenized, meaning they give their consent to be followed indefinitely, all the way to end-stage kidney disease as an example. And so some novel comparisons might be useful in that regard. I'll move on to TKV next, and TKV currently is not an accepted or validated efficacy endpoint in and of itself. At least that's our belief right now. And but we do believe that TKV can be predictive of disease progression. We don't think there's a need to study cyst volume in particular. TKV should suffice, but we do understand that sometimes there are acute effects on TKV that need to be accounted for in the analysis. It may be particularly useful as an endpoint for short-term proof of concept or dose ranging studies. And we believe that the agency, at least the FDA, has taken a stance where TKV may be a reasonably likely surrogate for meaningful outcomes, but probably needs to be complemented by measurement of other outcomes as well. In terms of the effect size that we think has reached a standard, we think that a minimum reduction in TKV growth that is likely going to confer a benefit is going to be in the range of about 25% when you account for chronic effects, or 50% acute plus chronic, which was the case for tolvaptan over its initial 3-year study. We know that it's a indirect measure, and as I said before, needs to correlate with GFR response. Some had hoped that TKV might eventually be able to be extrapolated to, or the reduction in TKV growth might be able to be extrapolated to a projection of prevention of dialysis or the time saved in dialysis, but this is going to be challenging. There are a lot of peculiarities and specific issues with the measurement of TKV. We know that measuring a patient at a single time point or even over multiple time points is less meaningful than looking at the population average. So it really does require that you look at a large number of patients, but look at each patient individually and their change from baseline as the key endpoint or estimand or variable that's going to be studied. And we would encourage that people use the best technologies, including centrally read within patient comparisons, to understand what the drug effects on TKV may be. With that, I'm going to hand this over to Ron to talk about the other category.
Thank you, Frank. So I'm going to focus on the endpoint variable of other. So as you know, most ADPKD subjects experience complications other than chronic kidney disease, although end-stage kidney disease is certainly a foremost concern. For example, hypertension, acute or chronic pain, and proteinuria, although this is typically mild, are issues that sometimes are bothersome or concerning in PKD. These have been used as components of composite endpoints, but only hypertension has been validated as a clinically relevant biomarker and has been well received. Secondary measures may focus on health-related quality of life and physiologic, chemical, or radiological biomarkers of disease progression or processes. Some of these include automated or ambulatory blood pressure measurements for hypertension, or the use or dosage of antihypertensive medications, endothelial dysfunction, and diastolic ventricular volume. As you know, the ADPKD impact and pain scales are not yet qualified, that is formally approved for use by regulators. Other potential secondary endpoints relate to bold MRI, renal blood flow, 3D ultrasound, which has been used in pediatrics, elastography as a measurement of fibrosis, and texture analysis as a way of getting more insight into the structure of cysts and kidney parenchyma. Finally, maximum concentrating ability as measured by urine osmolality, macrophage chemoattractant protein 1, hyperfiltration, serum copeptin, and FGF23 have also been proposed as potential biomarkers. One of the main points regarding the biomarkers is they've not been validated, the duration of impact is not known, and the range of variability is not known. So a lot of work remains to be done before we can use these other endpoints in clinical trials. So I'm now going to hand the floor back to Frank.
Thank you, Ron. This is our final slide, and it really focuses on interpretation of the study and the data. A number of these points have already been made, but we believe it's worth indicating that stratification is really critical. We know a lot more about PKD now and the covariates that are important. We also know about the effects of some therapies, but not all. For example, SGLT2 has not really been studied in ADPKD. And because of that, and because of its effects in general chronic kidney disease, we think that if it is being used by patients, that drug be stratified. We know that replication across different trials, different subpopulations, and different stages of ADPKD will be very helpful in helping regulators and clinicians understand the utility of drugs. We've mentioned the concurrence or alignment of endpoints is important. Dose response is typically done early because it's relatively ideal to use a smaller number of doses in a phase 3 program. And we believe enrichment really will facilitate the development of data for such studies and such drugs. But we want and would encourage companies to consider exploring broad inclusion-exclusion criteria to allow us to really understand the effects in patients outside of the enrichment population and potentially provide for broader utility of the agent in the real world. We think trials will be long and treatment effects off therapy may need to be examined and evaluated to exclude tachyphylaxis and other acute effects. And as we've mentioned any number of times, one must always plan a trial to account for or avoid missing data. Another consideration, and this is particularly important in pediatric populations, is compliance. We know that lifelong therapy, especially when begun at an early age, may be challenging. And we hope that the development of tools such as mobile device diaries will be helpful. One final question that we would ask for our regulators to comment on is what proportions of patients might be necessary for various ICH regions. We know that these are likely to be multinational, multi-regional studies. So it would be important to know if a minimum number of patients in the Japanese or Asian region versus Europe versus America would be important, as well as the minimal number of patients that are likely needed for safety evaluation for these drugs. So with that, we will hand over all these considerations and comments to the regulators. We'd like to thank everyone who has participated in the working groups. We love to thank the regulators who are going to provide us very robust commentary and dialogue during the subsequent sessions on and platform discussions. So thank you again.